Hello students, welcome to BioSir. I am here again with a new topic, enzymes used in recombinant DNA technology. Before going to the detailed discussion of today's topic, I want to just make some clarification here regarding my videos that you will not see any fancy animation in my videos, just I'll teach the topic, just I teach in my classroom. So please watch this video as you are attending your class and I guarantee you that your time will not be wasted. You will learn many new things and pure knowledge you'll get. So watch this video till the end and you'll learn many new things, I can guarantee you. And before starting the topic, I want another request to please subscribe this channel and like this video if you like my teaching. Okay, so let's start today's topic. So the detail I'll discuss today is a particular group of enzyme that is most essential in recombinant DNA technology, and that group is, together it is known as nucleases. There are hundreds of different enzymes which are regarded as nucleases, particularly depending upon their mode of action. They can be of two types, exonuclease and endonuclease, and within this endonuclease, the most important enzyme used in recombinant DNA technology or genetic engineering is restriction endonucleases. Okay, so I'll discuss in today's video these type of enzymes, and there are some other enzymes which are also essential for the modification or manipulation of DNA in recombinant DNA technology, and those are DNA ligases, DNA polymerases, alkaline phosphatase, polynucleotide kinase, and terminal deoxynucleotide transferases. I'll discuss these enzymes in my next video, not in today's video. Okay, so let's start. So before again knowing about details about this group of enzymes, let's first clarify the term enzyme. I guess that all of you have a basic idea what is an enzyme, but still I'm just making it clear to every one of you that enzymes are biological catalysts. Okay, biological catalysts, and they are mostly made up of proteins. Okay, so enzymes are the molecules, proteins, that act as catalysts to enhance the speed of biochemical reactions in living organisms or within living cells. And in that process, in that biochemical reaction, the enzymes are not used off or they do not get changed. Okay, they remain unchanged during the reaction and just simply they carry out some biochemical reaction. Okay, just enhances the rate of this reaction, enhances the rate. Okay, and they are highly specific for their substrate. Okay, so for any biochemical reaction there is a particular specific enzyme, and the enzymes are made up of mainly proteins. Okay, so again another information that the enzymes may be made up of a single protein. Only a single protein may be an enzyme, or in some cases enzymes may be made up of multiple proteins. Okay, here you can see that 3 different proteins constitute a single enzyme. In that case, it will be known as a trimer. Trimer or 3 subunit protein. Okay, trimeric protein. So depending upon the number of subunits there, regarded as trimer or tetramer or dimer, if 2 proteins are there, they will be known as dimer. Okay, tetramer, pentamer, hexamer. Different enzymes have different subunit composition. Okay, but all these subunits are of protein. Okay, protein. Clear? Just ignore my handwriting here because it is very difficult to write on the screen. Okay, in some cases, some enzymes have a non-protein component, which is the molecule is organic in nature, but it is not a protein component and remains loosely attached to the protein subunits and help the enzyme to carry out its reaction. So that is why they are known as coenzymes. Okay, and the non-protein organic molecules may be, in most of the cases, comes from vitamins like NAD+, or a flavin adenine dinucleotide, or in short it is FAD. Okay, so these act as coenzymes. In some other cases, some enzymes may also require some non-protein and inorganic ions. like Mg2 plus magnesium ion, zinc ion, particularly divalent cations. Okay, divalent cations and remain adhered to the protein subunits, and in that case they are known as cofactor. Okay, what I am saying this because the restriction endonucleases, which I'll discuss in detail today, restriction endonucleases are such type of enzymes where multiple protein subunits are there and they require cofactors and coenzymes during their reaction. Okay, so I guess now you can have you have some idea about enzymes. So now let's start about the main topic of today's video, that is nucleases. So nuclease is a group of enzyme which the main function of this enzyme is to break down nucleic acids. Actually, they break down either DNA or RNA. So then from the name itself, you can have an idea that nuclease means nucleic acid. This term nuclear is coming from nucleic acids. Okay, and enzyme suffix is used as is. I think you know that all the enzymes are named as a suffix is added at the end. That is, the suffix is. Okay, A-S-E, and plural is ises. Okay, so the main function of this nucleases is to break down DNA or RNA chains. You know, you have idea about the structure of DNA and RNA. DNA and RNA are made up of multiple nucleotides. Nucleotides joined with each other by the help of phosphodiester bond. Okay, so these are different nucleotides. Guanine is here, ribose sugar, this nitrogenous base, and this is the phosphate group. So here adenine is there, ribose sugar, and this is its phosphate group. So the phosphate group of one nucleotide, phosphate group of adenine remains attached through the 3' hydroxyl group here of the previous guanine residue, also previous nucleotide. So this bond, this covalent linkage between the 3' hydroxyl group of the previous nucleotide and the 5' phosphate of the next nucleotide is known as phosphodiester linkage. Phosphodiester. So here, two ester linkages are there involving a phosphate group. Okay, so that is why it is phosphodiester linkage. So the main function of this group of nucleases is to break this phosphodiester bond. That is why the DNA or RNA chain becomes fragmented. Okay, so depending upon their function, their mode of action, the nucleases can be of two different types, exonuclease and endonuclease. So exonucleases can break the phosphodiester bond of terminal nucleotides. Here from this figure you can see that this is one of the strand of DNA, this is another strand. So exonucleases can break the phosphodiester bond of the last nucleotide. This one is the last, this one is the last of this end, this is another last nucleotide, this one another last nucleotide of the other strand. Okay, so these last nucleotides can be eliminated. These phosphodiester bonds, this one or this one, this one or this one, can be broken by exonucleases. Okay, so if you incubate the DNA with the exonucleases, we ultimately will get single nucleotides in the solution. On the other hand, endonuclease, the meaning of the word endo means within. Okay, within. So the cleavage occurs within the within or endogenous phosphodiester bonds. Okay, internal phosphodiester bonds. Here you can see the arrow is shown here. So these are the internal phosphodiester bonds. So these bonds will be broken. So ultimately, small fragments of DNA or RNA will be produced. Clear? Okay, now let's see some examples of exonucleases and endonucleases. Exonucleases, particularly 3 different types of exonucleases are found in different living organisms. 5' to 3' exonuclease, which functions from the 5' end, 3' to 5' exonuclease, which functions from 3' end of the nucleic acid, and a poly-A-specific 3' to 5' exonuclease, which functions in the 3' end of particularly mRNA molecules where multiple adenine residues are there. Okay, so poly-A, wherever poly-A will be there at the 3' end of nucleotide, the exonuclease will cut those adenine residues. Okay, these are some examples of exonucleases that are used in recombinant DNA technology. First of all is BAL31, that is purified from the bacterium Alteramonas aspergillana and removes the nucleotides from both the ends. So BAL31, if we apply that enzyme with DNA, so it will digest both the strands from both the ends. So ultimately you'll get single nucleotides. Exonuclease 3 on the other hand, exonuclease 3 is purified from Escherichia coli bacterium. Okay, it degrades just one of the strand of a double-stranded molecule from 3' to 5' direction. So this particular phosphoester bond will be broken of this strand and this one will be broken of this strand. So one after another, these nucleotides will be eliminated from this end and of this strand, lower strand, this strand, it will be digested. So ultimately, you'll get short fragments of single-stranded DNA. For example, if you digest this one, and this one from this strand, and in the lower strand, this one will be digested, these 3. So these 4 nucleotides of lower strand and these nucleotides of the upper strand will remain in single-stranded condition. Okay, these remain undigested. Okay, so that is why you will ultimately get a short single-stranded fragments of the DNA. Clear? Another exonuclease is also used, that is lambda exonuclease, that removes nucleotides from 5' ends. Clear? Now let's have a look on some of the endonucleases that are used. The first one is S1 endonuclease, that is obtained from the fungus Aspergillus oryzae, and it only cleaves single strands. Okay, single-stranded DNA it can cleave. Here you can see from this example, this is single-stranded DNA, so single-stranded DNA will be digested. And in some cases, if there are some single-stranded stretch in a double-stranded DNA, for example in this figure, there is a single-stranded stretch here. 2 nucleotides are there in single-stranded state, so this particular position will be digested. Okay. The second one is deoxyribonuclease 1, or in short it is written as DNase1, that is obtained from cow pancreas, and it cuts both single and double-stranded molecules. Okay, DNase1 is non-specific in that it attaches DNA at randomly any phosphoester bond if broken. So DNase1 is active against single-stranded written is as SS, and in double-stranded dsDNA. Okay, ssDNA means single-stranded DNA and dsDNA means double-stranded DNA. So it is randomly can act in any type of DNA molecules. Okay. Now let's come to the most important enzyme used in recombinant DNA technology, that is restriction endonucleases. Okay, so why restriction endonucleases are known or named as restriction endonucleases, that we have to know first. Restriction because they are part of a particular system of prokaryotes. That system is known as restriction modification system, or in short RM system. So what is RM system? The restriction system, actually in 1950s, scientists noticed that the bacteria have the ability to restrict the infection of bacteriophages. You know that bacteriophages are viruses. Those viruses can attack a bacterial cell and can kill the bacterial cells. Okay, so in the bacteria, like E. coli, Escherichia coli, have the ability to restrict or inhibit the infection process of some bacteriophages. The mechanism was unknown, but that observation was there in 1950s. Okay, so later, 2 scientists, Arbor and de Siox, in 1962, actually discovered or clarified the molecular basis of that phenomena by observing that that bacterial cells have a group of enzymes. That enzymes can digest the viral DNA. Here you can see in this figure that viruses are inserting their DNA within the bacterial cell. Here the DNA is inserted. So after insertion of the DNA are recognized by restriction endonucleases in different locations, and the DNA is fragmented. So that is why the viral DNA is unable to replicate or proliferate into new viral particles. Okay, in this figure also you can see that this virus is inserting its DNA, but the DNA is immediately recognized by the restriction endonuclease, this yellow colored one. Okay, so this is digesting the DNA, so virus is unable to infect the bacterial cell. Clear? But here another problem was noticed by the scientists, that if the enzyme is able to digest DNA, so how the bacterium is protecting its own DNA? It is this particular, in this figure, it is shown the chromosomal DNA of the bacteria itself. So restriction endonucleases can cut the bacterial own DNA also, but that is not happening. So there must be some other mechanism by which the bacteria is protecting its own DNA, and that system is known as modification system. Okay, a modification system is nothing but having another enzyme that is known as methylase. Methylase. Okay, so what methylase does? Methylase adds a methyl group, CH3, methyl group to nucleotides in the DNA chain. So here in this figure you can see Me means methyl group. So methyl groups are added into the bacterial own DNA. That is why the restriction endonucleases are unable to recognize this methylated DNA of the bacteria. That is why they are unable to digest their own DNA. Okay, so that is why these two systems, there is restriction means the endonuclease function, and the modification means this methylase function. These two functions are integrated to each other to generate this restriction modification system. Okay, that part of a single system in prokaryotes. Okay, and there are three different types of restriction modification systems. Three different types. Those are type 1, type 2, and type 3. Okay. And among these three types, only the type 2 is used in recombinant DNA technology. Okay, so why type 2? Because type 2 restriction enzymes recognize and cut. They can recognize and cut the DNA at palindrome size with rotational symmetry. And different enzymes recognize different sequences. Clear? So what is a palindrome? The type 2 restriction endonucleases do not cut DNA randomly in any position. They can specifically bind to some designated places. Each of the enzyme have their own designated place, designated sequence where they can bind and can cut the DNA. And that particular sequence where it binds has a particular symmetry, and that symmetry is known as palindrome with rotational symmetry. Okay, so what is palindrome? I guess all of you know what is a palindrome. It is a word or phrase or sentence that reads the same backward and forward. Okay, if you read the word or sentence from the forward direction and backward direction, it will read the same. For example, here, madam reads the same if you read it forward or backward. Malayalam reads the same if you read from forward or backward. Here, one sentence is shown as example. Was it a car or a cat I saw? So if you read this sentence from forward direction or in backward direction, it will read the same. So this type of words are known as palindromes. Okay? But the DNA sequence is not exactly palindrome. It has a rotational symmetry. Okay. So here one of the example is shown. You can see here the DNA sequence is GAATTC. In one of the strand, it is GAATTC. And the sequence will read the same if you read from the opposite direction, but of the opposite strand. Not in the same strand. The sequence is not the same within the same strand. But the sequence is same if you read in opposite strand. Okay, it's complementary strand. So GAATTC, the sequence is same, but in opposite strand. Okay, and there is a particular axis of symmetry. If you see, this is the axis, the middle point of this 6 nucleotide sequence. So the sequence in this particular region, that is upper left side, will be same of the sequence in the lower right side of this axis. Okay, but in opposite direction. Clear? Likewise, the sequence in the upper right side will be same in the lower left side, but in the opposite direction. Clear? This type of sequence is known as palindrome with rotational symmetry. Clear? And all the type 2 exonucleases, there are thousands of restriction endonucleases that is under type 2, and each of these enzymes have specific sequence. This sequence is for a restriction endonuclease, which is known as EcoR1. Okay, we'll see many more examples in later part of the video. Clear? So why this type 2 restriction endonucleases are used in genetic engineering and not type 1 or type 2, type 3? Because they have some advantages. What are those advantages? The first advantage is that they have restriction and modification systems with different enzymes, separate enzymes, okay, and they can be separated from each other. So restriction and modification system was there, which both these functions are carried out by two different enzymes, and in absence of the modification system, restriction endonuclease activity can be done, which cannot be done with type 1 or type 3 restriction endonucleases. Okay, so what is the importance of this advantage? That modification in genetic engineering, modification is not required because methylation is not required for DNA manipulation. But restriction endonuclease or cutting of the DNA is very important. Okay. The second point is that the restriction activities, particularly restriction endonucleases, do not require any cofactors, coenzymes such as ATP or S-adenosyl methionine. Okay, those are required by type 1 or type 3, but type 2 does not require any ATP or S-adenosyl methionine. Instead, it requires Mg2+. Okay, divergent cation is required. Okay, and the third and the most important of all these advantages is that type 2 enzymes recognizes and defines easily symmetrical sequence and cut only within it. Okay, so a specific sequence is recognized and it cuts only within this sequence. Here the EcoR1 cuts in this position, in this phosphodiester bond between guanine and adenine in the upper strand, and in the lower strand also in between guanine and adenine. Okay, this position is cutted by EcoR1. But type 1 and type 3 restriction endonucleases digest the DNA randomly in any location. They recognize a particular sequence, but their cutting site is not specific. They randomly cut in any position of the DNA. Clear? So these are the advantages. Now let's see some of the examples of DNA sequences, some of the examples of restriction endonucleases that cut a specific DNA sequence. Okay, here you can see that thousands of more than 3,000 restriction endonucleases are known till now, have been isolated and purified from different bacteria. Okay, and the most widely used restriction endonucleases in genetic engineering, normally used in laboratory, are EcoR1, BAMH1, HIN3. These are HIN1, okay, SAL1, SAC1. There are hundreds of others which are not mentioned in this list also. Okay, so you can see all of these enzymes are isolated from some bacteria, like EcoR1 is isolated from E. coli, Escherichia coli, BAMH1 from Bacillus amyloleucofaciens, HIN3 from Haemophilus influenzae, okay, and these are their cutting sites, recognition and cutting sites, okay, and they either makes a sticky ends or blunt ends. Okay, we'll learn all these things in later part of this video. Okay, so according to their cutting site or recognition site, you can count these nucleotides 1, 2, 3, 4, 5, 6. So 6 nucleotide cutters are known as hexacutter. The sequence may be a pentacutter, means 1, 2, 3, 4, 5. Pentacutter is HINF1. Okay, it may be tetracutter, like SAU3A, ALU1, TAC1. These are tetracutter. There is octacutter also, but the most common type of restriction endonucleases are these hexacutter. Okay, and they are also used most commonly in laboratory. Clear? Hexacutter and sticky end cutters. Clear? These are some examples. Now let's see what is a blunt end and what is a sticky end. Okay, so restriction endonucleases can cut in specific locations, as I have told in the earlier slide. Again, depending upon their exact position in the nucleotide sequence, it depends that whether it will be a plant end or a sticky end. For example, you can see here that ALE1 cuts the DNA, both the strands of the DNA in exactly same location here, in this phosphoester bond between GC. Okay, so if it is cutted exactly like this, there will be no overhang. Both the strands will be of same size. Okay, there will be no overhang. So this type of ends are known as blunt ends, blunt-ended DNA. If there is no overhang of a single strand, this is known as blunt ends. But on the other hand, other enzymes, particularly hexacutters, cut the DNA, these two strands of the DNA in different position. For example, this strand is cutted in this position between G and A, and this strand, the lower strand, is cutted in this position. Okay, so this strand will be cutted like this. So there will be 4 nucleotide overhang. These 4 nucleotides will remain as overhang in the digested DNA. Okay, here you can see in this particular picture that these 4 nucleotides are overhang in the 2 fragments generated. So they are known as sticky ends. Clear? So depending upon the exact position of cutting, whether it will be a blunt end or sticky end, that depends. Okay, and there are some enzymes that generate same sticky ends produced by different restriction endonucleases. Their recognition site is different, but depending upon their cutting, same sticky ends is generated. For example, BAMH1, you can see BAMH1 recognition site is GGATCC. Okay, double G ATCC. So it cuts the DNA in this location. Okay, so fragment will be generated GATC. So this GATC is overhang. Whereas BGL2, another enzyme which has recognition site AGATCT. Okay, so AGATCT. This is the cutting site of BGL2, but it cuts in this location. So the same sticky end is produced. Its recognition site is different, but the sticky end is same. GATC, here also GATC. Another enzyme SAU3A, which is a tetracutter, okay, and cuts the DNA before that guanine residue. It recognizes this 4 nucleotide, that is why it is tetracutter. Okay, so it cuts before the guanine residues, and the sticky end is generated where also the sequence is same, overhang sequence is same, GATC. Clear? And most of the endonucleases, almost 99% of the endonucleases produce 5' overhang. Here in these examples also, the overhang is 5'. Okay, and there is some exceptions, like PST1, okay, obtained from Providencia stewarti, a bacterium, which cuts DNA with 3' overhang. Clear? So this is its recognition site, CTGCAG, and it cuts, the restriction endonuclease cuts the DNA in this position between A and G. So that is why a 3' overhang is produced. Okay, and rest all the restriction endonucleases produce 5' overhangs. Okay? Okay, next let's learn some other terms. Isosygomers. What are isosygomers? Now, there are several enzymes which have same recognition site and same cutting site. Okay, so isosygomers are those restriction enzymes which have same recognition site as well as same cutting site. For example, EcoR1 and FAN1. Its recognition site is GAATTC, and both the enzyme cuts in the same location between the G and E nucleotide. Okay, so those are known as isosygomers. Neosygomers, on the other hand, have same recognition site. Their recognition site is same, but cutting site is different. Let's see some example. Here, SMA1, SMA1, its recognition site is CCC. XMA1, its recognition site is also CCC. but the cutting site is different. You can see here, it cuts, small one cuts in the middle position, so it will be a blunt end cutter. Okay, whereas XMA1 cuts between the two cytosine residues, so it will be a sticky end cutter. Clear? Nested sites. What is nested sites or nested restriction endonucleases? Nested sites means the recognition site of one enzyme will be within the recognition site of another enzyme. Here you can see one example, hint 3. Recognition site is AGCT, and ALU1 have recognition site of AGCT. So it is a tetracutter. AGCT, you can see here that AGCT is within the recognition site of hint 3. Okay, so that is why this ALU1 site is nested within hint 3 site. Another example, BAMH1 and SAU3A. SAU3A is also a tetracutter. GATC sequence is within the cutting site or recognition site of BAMH1. Clear? So this type of restriction endonucleases are called nested restriction endonucleases, and their sites are known as nested sites. Clear? Now let's see another important information, that what is the frequency of recognition sequences in DNA molecules? After how many nucleotides the recognition sequences, recognition sites of and the restriction endonucleases may be there. Okay, there is a general formula that the sequence, if the 4 nucleotides are distributed randomly, then the sequence will arise every 4 to the power n nucleotides. 4 to the power n, this is the formula, okay, where n is the number of nucleotides in the recognition sequence. So if it is a tetranucleotide like GATC, it should occur once every 4 to the power 4, that is 256 nucleotides. So every 256 nucleotides apart, one sequence will be there. And if it is a hexanucleotide, a hexacutter like this one, so the frequency will be every 4 to the power 6 nucleotide equals to 4096 nucleotides. Clear? And this is applicable, these calculations are applicable if the nucleotides are ordered in a random fashion and that the 4 different nucleotides are present in equal proportion. Clear? Then only this is possible, this calculation is possible. But in reality, many of these recognition sites occur less frequently. Okay, in reality, it is not observed, it is not found that the restriction sites are these many nucleotides apart. Okay, many less frequent occurrences are observed. For example, the lambda bacteriophage has a genome size of 49 kilobase pair. 49 kilobase pair means 49,000 base pair. Okay, so if it is a hexacutter, if it is, for example, if EcoR1 is a hexacutter, 6 cutter, so every 4096 nucleotide apart, one cutting site will be there. So theoretically, in 49 kilobase pair of DNA, at least 12 cutting sites may be there, theoretically, but it does not found. For example, BGL2, which is also a 6 cutter or hexacutter, have only 6 cutting sites. Okay, BAMH1, which is also a hexacutter, so it should have also 12 cutting sites in the genome, but it in practically it has 5 cutting sites. SAN1, which is also a hexacutter, so it has only 2 cutting sites. Okay, so theoretically it can be calculated, but practically it varies. Clear? Okay, let's learn another thing, that how these restriction endonucleases are named. How the naming is done. Every time a new enzyme is found, new enzyme is discovered, so there must be some name of the enzyme, and that naming is done by following some rule. For example, you can see here, SMA1, HE3, hint 2, hint 3, BAMH1, EcoR1. All these enzymes are named based on the bacterium from which it is obtained. For example, EcoR1, from the name itself, you can have idea, you can guess that E is coming from the first letter of the genus name, Escherichia is the genus name, and coli is the species. So first letter of the genus in capital and the first two letter of the species, co. Okay, it is in small letter. So first letter will be in capital and two letter coming from species will be in small letter. And R is the strain name, okay. So in bacteria, a single species may have multiple strains. So each of those strains are also named, and the strain is taken in the naming of restriction endonucleases, okay. And it is written 1, 2, 3. These are written in Roman 1, Roman 2, or Roman 3, or Roman 4, 5. Okay, and this number is the serial number of its discovery. So EcoR1 is the first restriction endonuclease isolated from Escherichia coli strain R. Likewise, SMA1 has been isolated from Serratia marcescens, S capital letter and MA small letter, and in the first enzyme. It is the first enzyme from this in this bacterium, okay? HA3 from Haemophilus H and AE, Haemophilus, and it is the third enzyme. Clear? Hint 2 and hint 3, Haemophilus H influenzae IN, and strain, this D, this hint D is coming from its strain name. Strain name was D. Okay, so this one was the second enzyme and this one was the third enzyme from the same bacterium. So that is why they are named like this, hint 2 and hint 3. Okay? Okay, now let's see how the restriction endonuclease restriction digestion of DNA is performed in laboratory. Okay, so then you have to make a solution. You have to carry out the reaction in a particular solution where you have to mix the DNA you want to cut or you want to digest, okay, and the restriction enzyme, okay, and a buffer in which the restriction endonuclease performs its function optimally, and sterile water to make up the volume up to desired volume. For example, 20 microliter reaction mixtures are prepared. So for this, the DNA is needed up to 5 microliter. And as the 20 microliter of restriction and reaction mixture is there, so buffer will be taken 2 microliter so that the final concentration becomes 1x. The buffer normally remains in 10x concentration, means 10 times higher concentration than actual required. Say 1/10 of the volume is added so that it becomes 1x or the actual concentration. And the restriction endonuclease, generally 1 unit of restriction endonuclease is given. Okay, so what is 1 unit of enzyme? You should also know this particular concept. What is 1 unit of enzyme? It is defined as the quantity of the enzyme required to cut 1 microgram of DNA in 1 hour in optimal condition. Okay, so the amount of enzyme that will be required to digest 1 microgram of DNA in 1 hour time in optimal condition will be known as 1 unit of enzyme. This is the component of the buffer shown. This is one of the example where HCl is there, pH 7.4. Magnesium chloride, as I have told that magnesium ions are required for the restriction endonucleases. So magnesium salt is there, NaCl is there, and dithiotretol is there, DTT. But this constitutes the 10x buffer. Okay, these constituents are added one after another, and after digestion for 37 degrees Celsius for 1 hour, or this temperature may vary depending upon the enzyme used, because each of the enzyme has a different optimal temperature. So depending upon the optimal temperature, this incubation temperature will depend. Okay, after 1 hour so incubation, the reaction is stopped. The reaction is stopped by adding phenol or EDTA or heating. Okay, any one of these can be used to stopping the reaction. After 1 hour of incubation, this reaction has to be stopped. So that is why this step is done, and ultimately digested DNA fragments will be obtained. Okay, so I hope all of you have learned a lot. I guess I have been able to give some new information to you, new knowledge to you, and I hope all of you have enriched. So lastly, I'll again request all of you that if you have liked my teaching, like this video and subscribe my channel. And thanks for watching.